It's now my pleasure to introduce our next speaker. Our next speaker is Dr. Walter Koroshetz, who's the current director of the National Institute for Neurological Disorders and Stroke. And I promised to keep my introduction short, so I'm going to do that. Dr. Koroshetz got his MD at University of Chicago and then trained in both medicine and then neurology at Mass General Hospital at Harvard, and remained at Harvard for the next 20 years, where he did basic research, clinical research, and was a really important part of the clinical team there, involved in training residents as head of the residency program, and starting up stroke and neurocritical care services there. In 2007, he then left MGH to go to become the deputy director at NINDS, and then later the director. And I think that his experience in all of those clinical and research spheres really helped NINDS bring a lot of initiatives forward once he arrived. So he was instrumental in starting the Office of Clinical Research and Clinical and Translational Research, also NeuroNext, which is a clinical trials network for neurological diseases, StrokeNet, which is near and dear to my heart, which is, perhaps some of the work we heard about today might someday be tested. It's a national set of sites that can perform clinical trials for stroke and stroke recovery. And then finally, he's been instrumental in starting an R25 training program, which helps create physician scientists. It helps neurology and neurosurgery residents get additional training in science to help them do rigorous and exciting work going forward. And there are many more things I could say, but with that, I would like to welcome Dr. Koroshetz to give his talk, which is the Golden Age of Neuroscience.
Thank you so much, Marion. And I could share my screen there. Is that good? Great. Well, it was a real honor to be able to talk to you guys today. And so I'm going to talk to you a little bit about my look from 30,000 feet at what's going on in neuroscience. And again, it's only one person's look, so take it with a grain of salt. Then I'm going to give you some homework to try and think about some of the things that are worrying me, and then end up with a presentation of what I think is really exciting work that's going to completely change neuroscience now and in the future. And I'm not joking when I make the title The Golden Age of Neuroscience, because I think that's really what we're entering right now. And I just go back to when I was a kid, and I remember, the famous physicists of the early 1900s, and here they are sitting around, all guys except for Madame Curie, if you noticed. So that's going to change. I think we have equivalent breakthroughs that are going to kind of give us insight into our own brains, how our own brains process information, and that's going to lead to a tremendous ability to treat disorders of the brain. Now, that is fueled by lots of funding. So I want to just emphasize to folks that neuroscience at NIH is incredibly healthy. As a matter of fact, neuroscience gets more funding than any other area of science, and that includes cancer and infectious disease, although with COVID, that might change for a while. And the second point is that there are many institutes at NIH that fund neuroscience. NINDS is 100% neuroscience, but many other institutes have major components, particularly now the National Institute of Aging, which is budget is increased by $2 billion a year for research into neurodegeneration and dementia. So it's very healthy from the funding standpoint. And I want to mention, and probably the people here know, there's a considerable amount of funding that moves from NIH to Stanford. So our folks estimated the NIH Stanford funding at $757 million a year, and the NINDS funding at about $58 million a year. And some of our most high-profile, large team projects are being run out of Stanford, which some of them are mentioned here. And there's many, many, many more. So I guess the point to make is that from NINDS, we actually fund the research. I don't actually do anything anymore. And so I'm incredibly appreciative of the people who do the work, many of whom are at Stanford. Now, the funding is going to neuroscience because I think people are attracted to try and understand how our brains work. I think that's something that's hardwired inside our brains, actually. But that's not why the taxpayer is investing in neuroscience. The taxpayer is investing in neuroscience because diseases of the nervous system are the leading cause of disability in the United States. So you can see here the neoplasms are at 14, cardiovascular disorders, which include stroke, probably at least 40% of that is stroke. And then you see substance abuse disorder, neurological disorders, mental health disorders, and pain coming in and forming really the bulk of the disability in the United States. So although we have the investment coming, the responsibility is to make a difference for these disorders because we're not the National Science Foundation, we are the National Institutes of Health, and the taxpayers' funding is coming to us to understand the nervous system and then to use that knowledge to better treat diseases. And in fact, that's really what our mission is here at NINDS, is to seek fundamental knowledge about the brain and the nervous system, and then to use that knowledge to reduce the burden of neurological disease. So how do we do that? And we'll talk about that in a little bit of detail coming up. But the first thing is we invest across a long spectrum of basic translational and clinical research. What we don't want to have happen is have little gaps that don't allow discoveries to come to translation, and then gaps that prevent translational work from actually coming to patients. So we try and invest across that whole spectrum, although as you'll see, we don't have any, not nearly what I would say, it's not an equal distribution across. We're heavily invested in the basic side. Next, we're also looking always to find gaps in research and public health needs. And one of the gaps which we'll be talking about is the gap in the neurotechnologies that were available just 5 to 10 years ago for us to understand how the brain processes information. And the Brain Initiative has really been an amazing filler in of that gap, and we'll talk about that. And it has done that because development of tools and resources is incredibly important. One of my understandings is that we are no smarter than the people who came before us, but we have much better tools than they did. And the next generation is going to have better tools than we do. And that's really what drives a lot of the discovery, is the ability to have those tools. And then, as we're doing now, communicating, collaborating with stakeholders, including the public, is a part of our mission. And it's a two-way thing. We want to let people know where their money's going. But here at NIH, the danger is being isolated in a silo and not really in communication, not listening to what's going on in the community. So that's why I'm going to give you some of your homework, which is to try and help me with some of the problems that I think we're facing. So a two-way communication there. And we're always trying to continuously improve. We're in the middle of strategic planning now, lots of work going on to give us advice on how we can be a stronger institute, and we do that continuously. And then to mention that we are really invested in training. As I mentioned before, the next generation is going to have better tools, they're going to have better abilities to answer questions that the current generation is knocking their heads against the wall with, but we need these new scientists to come. And clearly at NINDS, it's long been a mission to improve diversity of our workforce. We've put lots of real clever folks thinking, funding to try to do that. And we have not come close to the successes that we wish. So if you look at our R01 portfolio, which is the kind of the standard grant at NINDS, only 1% goes to African Americans, a little more to Hispanics, and that's despite years and years of work. So there are things that we're trying to do. We have a suite of programs in training to improve diversity, which you see on the bottom of the screen. And I won't go through all those, but they all have these letters and numbers after them. So I would encourage people, you can put these slides out to folks and they can Google what do these letters and numbers mean. And the green at the top are programs that are particularly for physician scientists We'll talk about that in a second. We really need help in this space in trying to bring in new people into neuroscience, particularly physician scientists, and particularly people from diverse backgrounds. Now, I mentioned right from the beginning about the spectrum of research, and now you can see that pipeline. So on the basic side, we fund research that's what I call disease agnostic. It's about learning how does the brain work, how does the nervous system work, the spinal cord, the peripheral nerves, the autonomic nervous system, enteric nervous system. And it's that foundational knowledge that allows us to better understand disease. When you look at where our problems are when we come to disease, it's usually because we hit a wall where we have no idea about the basic biology. One example of that, is ALS, where, a lot of the genetic abnormalities start pointing to nucleocytoplasmic transport, not something that one would intuitively have thought. And learning about how that happens in a neuron we're starting from ground zero. So it's these fundamental gaps in our knowledge that prevent us from being successful as we move along. I often use the analogy, it's like crossing a river and you can see the stones going out into the river. And the first two, you can say, I can jump that. But then as you get further on, the stones get further away and it gets pretty clear that, you can try, but likely you're going to fall in the water because that gap is just too wide. And that's what we're up against with our really infinitesimally small knowledge about how the nervous system works when you compare it to what would be a full knowledge. But most of our research, 65 to 70%, goes into this basic research area, which is also mechanisms of disease research. And some of that involves patients. There's basic research trying to understand disease processes in patients. It's not just in a dish. Now, to move these to actually make impact on health, one has to move along the process to develop treatments, and that gets you to commercialization at some point, or else you get nowhere. So we have at NINDS a group, which is kind of like a virtual pharmaceutical company, a translational program that can help investigators from academics and small biotechs kind of move through that pipeline to get their discoveries to the point where they can get an IND or an IDE from the FDA to go into patients and test it. And we're not in the business of making drugs and selling drugs. Our business is really to de-risk this process, to bring the process to the point where a company can say, that looks like it could work, and take it off our hands. And unfortunately, for many of our disorders, that's a problem, that many of the companies don't want to go near neurological disorders because of all the failures in the past. And the poster child for that is stroke, where there's really hardly any companies at all that are interested in stroke after the failures of the late 90s and early 2000s. And then we have a division of clinical research, which is really expert in doing clinical trials and some of the networks that Marion talked about earlier. So if we go back to our basic research, it really spreads across the entire nervous system. And we have these little groups of program directors, and we have one or two program directors for each area of science. And if you want to understand how to negotiate the NIH system, what you do is you find out who's the program director that fits your science and email them and develop an informational relationship going back and forth, because their job is to help you try to access the funds for your passion, to follow your passion in science. Now, here's your homework. I just wanted to throw out a couple of things that are really bothering me, and hopefully that you can help come up with solutions. The first one actually came from Tom Sudoff, who's at Stanford, and he wrote a piece in Neuron saying that molecular science is really not getting the kind of respect that it deserves, that it's really become subservient to systems neuroscience. As a result, the research into biological processes is increasingly limited to disease-related events. Molecules are primarily considered as tools, and fewer fundamental questions about how the brain works are addressed. And if that's true, that's going to be dangerous. Now, there are certainly swings in science, but I'd say that Tom's warning is a good one. The last half a century has been dominated by the molecular neuroscience. So I have here a list of all the neuroscience Nobel Prize winners from 1936 to 2014. As you can see on the left, it's been really dominated by these amazing discoveries on the molecular components of information processing in the brain. And the systems neuroscience, with the exceptions of Huber, Wiesel, and Sperry, and Moser, have really kind of lagged. But as Tom mentioned, and as people at Stanford probably know, and we'll talk about coming up, this is changed because the tools to study systems neuroscience and to understand information processing on the system level have really improved. And that's certainly bringing a lot of people into that area of neuroscience. But the molecular neuroscience is equally important. The benefit is the convergence of these two streams of science, where you can understand how the molecules are working at the cell level, at the intercellular level, and then use the systems neuroscience to understand how manipulations at that molecular level really affect the circuit system. And that's something that I think is really where the golden age will be achieved when we can converge there. The second problem that we're facing is what we call the big data problem. And I show here on the left one of a replica of Guggenheim's first printing press, and a paper from 1645, one of the first philosophical collections, to make the point that the way we share our data is really in publications not much different from 1645. And yet we have the ability now with these new technologies to bring data in a different manner, and how that's going to sort out, I think, is a really important question, and will change how science is done. As one person wrote, a paper really has its science confined to the methods and the results section, and everything else is an advertisement. And so how to move that data and methods into platforms that are much more conducive to promoting the science enterprise is a big question. And we have tried in multiple different areas, which I can show, which are showed here. We have set up really, first-rate data platforms for traumatic brain injury, for Parkinson's research, for the HEAL Pain Initiative, for the Brain Initiative, but they're incredibly expensive, and they don't generalize across all of what we do. And so they're not really this is not a sustainable system right now. We need better ways of bringing data together, making it useful, not collecting data for the sake of collecting it, but collecting it so it's useful. The third problem that I think we're facing is that science always kind of defaults to the easiest model system, easy meaning most likely to give you an answer. And that's a good thing. But generalizing that to the human nervous system, there's a lot of stones that you'd have to jump across to get there. And so our predominant focus on the mouse, I think, limits us to look at really how the nervous system sits inside the body, and how it's influenced by the vascular system, by the CSF, the meninges, the choroids, the lymphatics, and then the autonomic nervous system, the enteric nervous system. Lots of research in the space, in the 50s and 40s, not much now anymore. And so I think that's also limiting, and we're missing a lot of the inputs to the brain, either through immune interactions. Even now, the gut, for instance, is clearly incredibly important in influencing brain function, potentially through its immune cells in the gut that traverse to the brain. Parkinson's disease, there's a pretty good evidence that it may start in the nerves to the gut and move into the nervous system. So, and yet these areas have been so neglected that when you start to study these things, say in Parkinson's, you're also starting from ground zero again. And the last one I wanted to throw out, which is similar, is the need to look at animal models when it's necessary, because the mouse just isn't going to do it. And there are certainly lots of behaviors, there's lots of circuits the mouse just doesn't have. And so we need to really redouble our efforts to look at non-human primates when it's appropriate and do it in an ethical manner. But this is something that is certainly under siege in the US, and we will pay a severe penalty if we can't come to grips with this problem. Now I'm going to jump into translational resources for a second. I mentioned we have this virtual pharmaceutical company, and they have multiple programs, again, as I mentioned, to bring things to the point where they can go into patients. And so if people are interested in translation, we have these programs. We have a course, we have training programs to get people interested in the science of translation. Lots of scientists think they made this great discovery and they can cure things, but they have no clue how to go about the process to get to the point where you can actually give something safely to patients. And this is something that is, it's like any other science. One has to learn it well to do it well. We have also kind of approached the biomarker problem in a similar fashion, really putting rigor into not just, finding something that correlates with a disease, and publishing it, but then going to the point where it's validated and it's going to be useful in a phase 2 trial. And so we have a biomarker program that's almost like a drug development program in terms of its milestones. And it's all really focused on developing signatures that can be tested and help phase 2 trials to know if you're hitting your target, if there's proof of principle. And the advances are coming. Now, it's never know when they're going to come, but here's one that is really spectacular, the Rosin treatment for spinal muscular atrophy. We worked here at NIH for years on a program to improve the production of an alternative protein that would ameliorate this disease that kills young children. Before they're 1 or 2, it's almost like ALS in children. And then the discovery of antisense to prevent to enable that SMN2 protein to be produced in greater amounts has really been a miracle, that children who would previously have died are now walking seemingly doing really well years and years after the beginning of treatment. So at tremendous effect sizes when you hit right in the middle of the target of a disease. And this disease, spinal muscular atrophy, there are hundreds of diseases that are similar. And so we are starting up processes to open up this and streamline the process of getting gene therapies tested in multiple rare disorders. On the clinical side, we have research to get what we call readiness for clinical trials. So to study a population, validate a biomarker, get things ready to do a clinical trial. And then we have a whole suite of different clinical trials, networks in different areas. The NeuroNext network, which Mary mentioned, is to do phase 2 trials in any neurological disease. It's disease agnostic, and it's been quite successful, and it's been much more efficient than what we did in the past. And here you can see the multiple sites across the country. Now, as I mentioned before, one of the major obstacles is the workforce. What I show you here is the number of K applications, K08s, K23s. These are the signature physician scientist awards given out by NIH. And as you can see, since 2000, it's basically flat. The number of medical school faculty has gone up. The number of medical students has gone up. The number of physician scientists has been flat, probably back into the 1980s. And why this is a real mystery to me, because particularly for neurology, combining research with clinical care, they seem so complementary, hard to do one without the other in my mind. And for NIH, for NINDS, we've been flat too at about 70 applications a year. So, and we fund about half of them. So this is the numbers. This is the bottleneck. If you're going to solve tough clinical problems, 50 people a year of funding is not going to help. Okay, it'll help, but it's not going to do it. Now I'm going to switch to the Brain Initiative, which, as I mentioned, is really one of the most exciting things that are going on at NIH. And this was a plan put together by Bill Newsom and Corey Bargman. And it's a focus on developing the tools. It's called Brain Research to Advancing Innovative Neurotechnologies. So developing the neurotechnologies that are focused on seeing circuits in action. Because all of our brain activity, all of what we can do is based on this brain activity, circuit function, information processing, and what is the most complex computing device ever imagined, with 85 billion neurons, similar number of glial cells, trillions of connections, and things happening in millisecond timescales. From the clinical standpoint, I'd say that, in neurology, we've looked at diseases based on the pathology. What does it look like when we look in the brain? That's kind of the beginning of neurology. And then treatments are designed to try to improve whatever the pathology is. And that's worked for some things. But for some things, when you look in the brain, you don't see anything. Tourette's syndrome, schizophrenia, there's no pathology to see. And actually, even when you see pathology, say Alzheimer's disease, it's not the amount of pathology that really matters. It's what the pathology has done to the circuit function. And the problem that we've been facing until now is that we can't see the circuit function. The tools we had, say like EEG, is a tool from the 1920s, and it's incredibly crude, and it hasn't really done what we needed. And functional MRI, similarly, the problems have been so severe that it's been around for 40 years and it's not clinically useful for almost anything. Maybe to look and see what side speech is on for intent and for testing people prior to epilepsy surgery. But we really need better ways to examine circuit activities. And so in the Brain Initiative, the pushes are to map, monitor, and modulate circuits. And we take our marching orders from Freeman Dyson, who said that new directions in science are launched by new tools, much more than by new concepts. The effect of a concept-driven revolution is to explain old things in new ways. The effect of a tool-driven revolution is to discover new things that have yet to be explained. And just in my career, so when I came out of medical school, this was the first scans of living people on the left. It's a CT scan. And basically you can see the black fluid-filled space, but otherwise not much of anything useful. This is where we've come in the years since with MRI scanning. And this has completely revolutionized neurology and neurosurgery, but has hardly any effect on psychiatry, which is making up a huge burden of brain diseases. Now, luckily, the Brain Initiative has been able to attract Dr. John Nye to come in as the director. I was co-directing it with Josh Gordon, and then before that with Tom Insel. But now we have a real full-time director, which is about time, and John is super. And I think we'll do a great job. I want to just talk a little bit about the Brain Initiative. There are basically 7 pillars, and the first of which is discovering diversity. What that means, in simple terms right now, is trying to identify all the different cell types in the brain. As I mentioned, there's 85 billion neurons, 85 billion glia in the human brain. So this is a challenge. But surprisingly, this has taken off. And that's because of technologies that allow high throughput studies of single cell RNA that you can then analyze and parse out the different cell types. And amazingly, which really was a surprise to me, is that you can get almost the same information by doing single cell analysis on the nuclei. And the reason that's important is because you can do that in human autopsy brain tissue. So this actually is giving us a cell census of the brain, but it's also revolutionizing neuropathology. It'll take time, but I think it will make neuropathology quantitative and precise. In terms of research, not only do you get the cell type, but from the transcriptome, you get the cell type specific transcription factors that'll give you cell type specific access to the cells with genomic tools. And there are many of those that have really changed neuroscience. Some of those tools have allowed us to label cells and see where they're connected. And so we can develop circuit diagrams that vary in resolution. Using electron microscopy, we can actually get incredible nanometer level connectomes of brain tissue now. And that's something the Brain Initiative is pursuing in a big way. We have tools now to monitor neural activity coming out of the work of Boyden and Deseroff at Stanford. Tools are now available where you can inject the gene into the brain of an animal or develop a transgenic animal, and those cells will light up when they become active. So in the past, you would have to stick an electrode into a single cell and record from a single cell. Now you can use this technology to record from millions of cells simultaneously and really see the network activity that we imagined was happening but could never see before. We can see the brain in action. And also these genomic technologies allow us to intervene with cell type specific precision to turn on or turn off cell types. And this is allow us to go past just the associative knowledge of one type of brain activity related to behavior, but actually testing the causality by intervening and seeing if the theory of how this is happening is really true. And the theory is incredibly important because we don't understand the language of the brain. We kind of know action potentials and synapse are important in communication between neurons, and that's kind of like the letters of the alphabet. But we don't know the words, we don't know the phrases, we don't know the paragraphs. And now with that network activity, we have the chance to do that, but it's a tremendous computational challenge to get there. But I think we'll eventually be able to make some inroads and almost come up not with Einstein's general theory of relativity, but some theory of general information processing in particular nuclei. And then this is also going to be incredibly important as we move to human neuroscience. And just a couple of examples. This is a paper that came out from Carl Deseroff and Ivan Soltes recently. And what they have here is they injected one of those genes I mentioned that only go into the parvalbumin cells, these inhibitory cells. And then when light hits these, the protein made by these genes, the cells are turned off. And so they use that to shut off a seizure, which is really way too robust synchronous firing. So here you can see in the gray, the seizure is going on. Here's a seizure that starts, they turn on the light, and the seizure stops. Now, the beauty of this is that they've developed this gene so that the opsin responds to a red shifted light, and you can actually get that through the skull of the mouse. And so this is actually turning on or off seizure activity from outside the head. Really amazing. Another example from Mark Schnitzer and Greg Scherer's lab at Stanford, where they found cells in the amygdala of the mouse. And these cells are involved in the pain perception, but they don't actually, turning them off really doesn't affect the ability of the animal to sense pain, but the animal doesn't care about pain. So if you can see here on this slide, here when they give the drug that activates this artificial gene and shuts off the cells, when the cells are shut off, the animal can go from the cold side to the warm side. It doesn't really matter. When those cells are active, however, when they don't shut them off, the animal will stay away from the cold side. It really doesn't like it. Here it feels the cold, it just doesn't care. What an example of something, if we could do this in people with chronic pain so that they can still get away from the hot coffee, but they're not going to be suffering from their pain from the low back. And this is just the last example.
Dr. Nader Pouratian implanted the device made by Second Sight over the visual cortex in Jason's brain. The Orion device converts images from a tiny video camera on a pair of sunglasses into a series of electrical pulses. Those pulses stimulate electrodes in Jason's brain that let him see patterns of light that act as visual cues.
So this device takes optical activity from a video camera and puts it right to the back of the brain where the visual system is. And this gentleman was blind due to problems with his eyes. And what he said was, when my wife was mad at me, I never knew because she's really quiet and I didn't even know where she was. But now with the device, I can see her. So he doesn't get a picture of the wife, but he can tell when she's there and when she's not there. And when she's there and quiet, she's not happy. Okay, now I'm going to end now by just mentioning the fact that this brain initiative and the tools that are coming out are becoming more and more powerful. We have the second part of the brain initiative just starting. A number of projects are on the verge of being launched. But what I wanted to end with is a message to people to start thinking about the ethics of what we can now do and what we will be able to do. We have, throughout the Brain Initiative, had a large focus on neuroethics. Hank Greeley at Stanford has been a real leader for us in this space. But we have to really think about the appropriate use of these tools. I feel very comfortable that on the medical side, we've been in this space before. And we've negotiated it, sometimes not perfectly, but ups and downs. We've come to the right space. But these tools have the potential to get outside of the medical space. And their society is going to have to make some real hard choices about what they would use to enhance, say, kids' ability to learn math. There's a circuit for learning math. When you go in, you don't have that circuit. When you come out, at least the school I went to, you had that circuit. So there will be ways to improve that kind of circuit behavior so that you can learn calculus. But there's lots of other things where the question is going to come up, is that really ethical to do? And as people may know, Elon Musk has a company that's done some really amazing stuff, building electrodes that are really way far advanced with the idea that these are going to be able to hook up to computers in his mind, millions of people. So this is something that really should be on the radar screen. And with that, I'd really like to thank you for your attention. Happy to take any questions.
Thank you so much, Walter. That was fabulous. I'm clapping. I'm pretty sure everybody else is. It's great to have an overview of everything that NINDS is doing, and it makes me really excited and hopeful about the future of neurosciences. We have a few questions now. I just want to remind everybody that there's a link you can click at the top of the video where you can enter a question that you'd like to ask Dr. Koroshetz. And I think there's a few there already, but I'm going to take the first one. I really appreciated how you talked about how important diversity is in the scientific workforce. I think it's something that's on all of our minds, and we really appreciate everything NINDS is doing to promote diversity. I'm particularly cognizant that when it comes to Black, Indigenous, people of color, that we have a really poor representation in the sciences. And the biggest drop-offs in participation are between undergraduate and graduate school, and then postdoc to faculty. And I wonder what you think universities can do best to help improve these statistics and make our community more inclusive.
Great. Well, I'm glad you asked. Just before this, I was on a meeting called the Endura Program, which we run out of the Blueprint for Neuroscience. That's all the neuroscience institutes. And that program is basically funds undergraduates, programs that target undergraduates to bring them into neuroscience. And that has been quite successful. 68% of the graduates of this program, and there's now, I think, almost 200, are in neuroscience PhD programs. And it just kind of hits the point that, you know, if you just think back at your own career, when did you make these kind of decisions on whether you're going to go to the NBA or the NIH, it's really, it's really at that college or high school level. Now, the Endura program works with universities and they reach out to undergrads. I don't see any reason why every university in the country can't do the same thing. We have about maybe 8 programs in the Endura. There could be 800. So I think that would be really helpful. And then in terms of the progression, we actually have grants. So we have a grant, a diversity grant for people to go from PhD to postdoc. So you get the grant, you'll have money to go to a postdoc. I'm sure they're going to love you if you come in with your own money. And we have another grant to go from postdoc to faculty. And then NIH itself is putting out this program, which I'm working on, called the FIRST program. And that's going to put money on the table for institutions to basically clean up their act, improve the culture at their institutions, but also give them money to hire cohorts of people from diverse backgrounds. The idea being, if you hire one, they really are standing out alone, and it's awkward, and they may not be successful, and their impact may be low. But if you can hire 3 people and they're a group, then I think that we're going to have much more success in changing the culture. So the first program is going to be rolled out. We're working on it now. Hopefully within a year or two, grants will be available.
Great, thank you. So the next question is from Bill Newsom. He's
He always asks the hard ones.
all right. So you want to take a deep breath and prepare yourself.
Okay.
Bill mentions that we're now about halfway through the BRAIN initiative, and that NINDS has been wonderfully supportive and effective at mining scientific gains from the initiative. And he wants to know if you have any knowledge of how the initiative is being received on Capitol Hill and what the prospects are for continuing it past 2025.
Okay, well, anybody asking about if I have any idea what's going on at Capitol Hill, boy. So that is a hard question, Bill. But the easy part to the question is that, prior to COVID, we had just a tremendous relationship with the people on Capitol Hill, both at the Senate and the Congress side. And I think there's a tremendous amount of enthusiasm for the BRAIN initiative on the Hill. The one caveat is that on the Hill, the BRAIN initiative is conflated with everything, every neurological, psychological problem you can imagine. So it's only a few folks that really understand the core feature, but they buy into the goal, which is to understand how the circuits work and to be able to come up with answers for the millions of people whose brains just are not functioning the way they want them to. So I think, we'll have to see after COVID. Right now, there's very little constructive conversations going on.
Yeah, unquestionably a difficult time for everybody. So we all hope that it will pass quickly. I think we have time for just one more question. So this is a question about the training initiative you have for physician scientists. And this is somebody asking about funding opportunities for translational neuroscientists with a PhD, but not an MD. Are there career development awards other than the K99R00? And what are you doing as an institution to help with training more translational scientists?
Well, that's a good point. So the translational part, well, I guess it's a little hard to know what they mean by translations, because it means so many things to other people. But
yeah,
when he was at Stanford, he had a program, if I remember right, for PhDs to actually work closely with the physicians to understand the diseases. I think that's a tremendously good thing to do. What we've done for the first time on the really what I call hardcore translation, which is therapy development, we have a course every year, which is very sophisticated, to try to teach people, goes on for about a week, to teach people what are the core components of doing translational research. And it's either out there or soon to be out there, a T32 program to do the similar thing. And we are trying to, as you know, Marion, we have these programs, career development programs for the K physician scientists. Steve Korn is also working to do that for our PhD grantees as well, the fellowship grantees. So I think that that's been a mistake, not to just give them the money and send them to the lab and say, good luck. So we are going to try to buff that up and try and really develop a community among the postdoc community that would be helpful in moving them forward.
That's great. Thanks for those answers and thanks for your talk.